morning, viewer. Welcome to our Glory Globe TV's amazing news. Depression. Yeah, depression is among the most widespread psychiatric disorders experienced by an estimated 3.8% of the global population. It is characterized by persistent low mood and a loss of interest in most activities, as well as possible disruptions in eating and sleeping habits too. The most severe and persistent form of depression, known as major depression, that MD, can be highly debilitating, often disrupting the lives of affected individuals and preventing them from working or engaging in other daily activities. Past studies suggest that depression is often caused by combination of genetic and environmental factors. The genetic risk of developing depressions or other mental health disorders is estimated using so-called polygenic risk scores, and these are estimates of an individual's predisposition to a specific conditions based on the cumulative effect of various genetic variants. Researchers at the University of Edinburgh, the University of Melbourne, Vrije University, Amsterdam, and other institutes recently carried out a large-scale study at exploring the relationship between PRS values for MD and people's brain structure. Their paper, published in Molecular Psychiatry, reports specific patterns in the volume and surface area of specific brain structures that are linked to a higher genetic risk for depression. The Neurobiological Association of Genetic Risks for MD remain underexplored in large samples with no comprehensive mega-analysis conducted to date, wrote Zhu Sen Yara, J. Twenders, and other colleagues. Our study analyzed data from 11 separate studies encompassing 50,975 participants from the NGMA Major Depressive Disorder Working Group. We developed highly consistent genetic and neuroimaging protocols and applied this throughout all participating studies, together with rigorous genetic methods to remove overlap between the polygenic risk scores, training, and testing sample. As part of their study, Sen, Twenders, and their colleagues analyzed a large amount of genetic and neuroimaging data collected as part of 11 international studies. Based on these data, they calculated PRS values that estimated the genetic risks that the study participants would be diagnosed with MD at some point in their lives. Finally, they examined the brain scans collected from the participants and tried to determine whether higher PRS values were linked to specific patterns in the volume of various brain regions. Behind this exclusive report.